The overall goal of this experiment is to detect changes in protein insolubility with age in C. elegans, and to evaluate how targeted gene knockdown influences this aging marker.
This method can help answer key questions in the field of aging and proteostasis, such as why organisms fail to maintain a healthy proteome with age. The main advantage of this technique is the opportunity to study protein aggregation during normal aging in the absence of disease processes.
Though this method can provide insight into protein aggregation in C. elegans, it can also be adapted to other model organisms, for example, to study age-related protein insolubility in mouse.
To begin treatment, add 207.45 milliliters of S-basal media with additional reagents as described in the accompanying text protocol to a 2800 milliliter Fernbach culture flask. Add a final concentration of 50 micrograms per milliliter of carbenicillin and 1 millimolar of IPTG, and close the flask with a membrane screw cap. Take the L1s out of the 25 degrees Celsius incubator and transfer them to 15 milliliter tubes. Centrifuge the L1s at 1900 times G for 3 minutes. After spinning, remove the supernatant. Under a microscope, count the L1s per 2 microliters and average the numbers obtained from at least 9 drops. Next, add 50,000 worms